Hello young guys, you are welcome in SOAP biology classes produced by Vidyarthi Pratibha Utsav. The director of this program is Mr. Pankaj Tiwari. Technical support by Rajnandini and Bhati Ratna. Resource person is myself, Dr.Agam Dayal. Actually, all scientific observations depend upon certain tools. So, every optical system has a limit which is called resolution limit. Resolution limit of human eye is 1 by 10 or 0.1 millimeter. It means that if anything is larger than 0.1 millimeter, then only our eye can see it. And if it is smaller than 0.1 millimeter, our eyes cannot see it. We can say it in other words, if there is distance between 2 points is more than 0.1 millimeter, they will appear two different points to our eye. But if the distance between 2 points is less than 0.1 millimeter, they will appear only a single spot by human eye. So the study of biology up to 1665 was only based up to morphology and anatomy level. But in 1665, Antony van Leeuwenhoek, a Dutch lens grinder, he had a hobby to make lens by grinding glass pieces, and he just made about 4,000 such simple microscopes. So magnifying glass is a simple microscope made up of only one lens. At this, he observed a drop of blood, a drop of water, a drop of semen, and many other things by this simple microscope. At the same time, Robert Hooke used a compound microscope made up of two lenses. So the magnifying power of simple microscope of Leeuwenhoek was 200 times. The magnifying power of Robert Hooke's compound microscope was about 2,000 times. Robert Hooke studied a piece of cork, cut it by a sharp knife, and observed under a compound microscope. He observed some honeycomb-like structures. In Greek language, kaitos means cell. So he named these compartment-like structures as cell. At the same time, Leeuwenhoek observed that many things have such structures. A Dutch scientist, Matthias Schleiden, said that all plant bodies are made up of such type of cells, and at the same time an English scientist, Schwann, said that all animals, the body of all animals are made up of same type of cells. This gave birth to cell theory. A very interesting thing is that cells are very small. Why they are very small? Because very small size have a larger surface area as compared to the volume. Suppose we have a cube having one side 2 millimeter long, 2 millimeter broad, and 2 millimeter deep. Then its total volume will be 2 into 2 into 2, 8 cubic millimeter. But the total surface area will be 2 into 2, because there are 6 surfaces. 2 into 2 into 6 will 24 square millimeter. If we cut this cube into 8 pieces from the middle, then each cube will be 1 millimeter long, 1 millimeter broad, 1 millimeter deep, there are 6 surfaces and there are 8 cubes, so the total surface area will become here 48 millimeters square, whereas the total volume is the same, 8 cubic millimeter. So this shows that as we reach up to smaller and smaller size of cell, the surface area increases, and that surface area provide a surface for entry of necessary nutrients into the cell, exit of waste products, entry of oxygen, exit of carbon dioxide, so it is much more beneficial. Second thing is that in our body many tissues are there. Tissues are groups of cells which perform same function. For performing different type of functions, we have different shapes of cells. For example, in our outer epithelium or inner epithelium, we have epithelial cells. Epithelial cell may be cubical, rectangular. Similarly, we have blood cells. RBC are poppin shaped, biconcave, very small cells. WBC are different shapes of nuclei. They are fighting against diseases. In our body, we have cartilage at some places. Cartilage have isolated cells with an intercellular matrix. Similarly, in connective tissue cells, the packing material, we have some cells, some intercellular matrix, and some type of fibers, elastin fibers and collagen fibers. The nerve cells are very much elongated for carrying the message. Our muscle cells have 3 types of muscles: striated muscles, which have many nuclei, cardiac muscles in the heart, which are branched, and then some smooth muscle cells found at certain specific areas of the body: mammary gland, wall of scrotum, wall of blood vessel, wall of alimentary canal. Similarly, this is chlamydomonas. Organisms may be made up of one cell, few cells, or many cells. Chlamydomonas is a green algae. It is made up of only one cell. That one cell is a complete organism. In our green leaves on the ventral surface, we have certain guard cells which are kidney shaped, surrounding the stomatal opening for entry of gases. Amoeba, a unicellular protozoan, the shape is irregular and it can form some elongations called pseudopodia in any direction for locomotion. All cells have some basic features which is called typical cell or a generalized cell. Let us see what is the structure of a generalized cell. So, normally a generalized cell contains an outer covering cell membrane and the inner content responsible for life, which is called protoplasm. In this protoplasm we have a central nucleus. The remaining part of protoplasm is called cytoplasm. But here at this stage we have 2 types of cells. One, a prokaryotic cell in which there is no nuclear membrane and the nuclear material DNA is attached with the cell membrane at a point. There are no cytoplasmic organelles, there is no histone protein. Such cells are called prokaryotic cell. For example, a bacterial cell. In a bacterial cell, we have a plasma membrane, then a bacterial cell wall made up of bacterial cellulose. There is a onion-like structure, mesosome. Its function is not clear. In some of bacterial cells, we have an extra ring of DNA which is called plasmid. There may be certain flagella. The ribosomes are scattered here in the cytoplasm. Then the other cells in which we have a nuclear membrane, they are called eukaryotes. And the eukaryote cells are found in all rest of organisms except bacteria. They are found in animal cell also, and they are found in plant cells also. In animal cells, this generalized animal cell is surrounded by a cell membrane, a nucleus, a centrosome. Centrosome is found only in animal cells, not in plant cells. In a nerve cell, neurons also, the centrosome is absent. So the nerve cells cannot divide. Then we have a structure called Golgi body, which is secretory structure of the cell for secretion of enzymes and hormones of need. We have certain lysosomes which can digest the food vacuum particles of the cytoplasm. If there is any broken piece of any cytoplasmic organelle, that can also be digested by lysosome. And if there is a need for digesting complete cell, it can also be done by this lysosome. So lysosome are also called suicide bags. Then, like a network of road in a city, here also all over the cell we have a network of double-walled tubules which are called ER or endoplasmic reticulum. Endoplasmic reticulum is of two types, some which have attached ribosomes, they are for transport of proteins, and some which do not have attached ribosome, they are called SER or smooth endoplasmic reticulum, they are for transport of lipids. Then in the nucleus is made up of two membranes, a nucleolus, there may be one or more than one nucleoli, and network-like structure called chromatin network. This chromatin network at the time of cell division breaks into separate thread-like structures called chromosomes. The number of chromosomes is fixed for all cells of a particular species. For example, in human being, all our body cells will have same 23 pair or 46 chromosomes, right? There is a mitochondria, which is the powerhouse of cell for oxidation of food material to liberate energy or oxidative phosphorylation. The plant cells have similar structure, but here a very big central vacuole is there, so the rest of the cytoplasm and nucleus, they are pushed on one side. Here the Golgi body is broken into many small groups called dictyosome. Both SER and RER are there. There is two special features. Plant cells have along with the cell membrane or plasma membrane, one more extra covering which is called cell wall, which is made up of cellulose, a carbohydrate. Then we have certain plastids. Plastids are found in plant cells only. There are three types of plastids. One, the leucoplasts, which are colorless, which are found in potato for storage of starch granules. Then in the leaves, in the green leaves, we have certain green chloroplast containing the chlorophyll molecule. In the flower petals, we have some colored plastids which are called chromoplasts. These chromoplasts contain carotene or xanthophyll. One more pigment is found, anthocyanin, in the cell sap of the vacuole. centrosome is absent in plant cells. The cell membrane is selectively permeable, which allows entry and exit of only a few types of molecules, and they do not permit some other molecules. They are living. But the cellulose cell wall is non-living. It is freely permeable for all types of molecules. So this much for the day. Thank you very much.